In 3 minutes, learn how PLA technology converts protein signals into DNA signals. The core principle is based on proximity ligation assay PLA technology. It is a next-generation, highly specific, highly sensitive, ultra-low volume, multiplex protein quantification technology that integrates site-specific and stoichiometric antibody-oligonucleotide conjugation, specific antigen antibody recognition, and qPCR amplification, providing an alternative to conventional ELISA. An enzyme-mediated method is used to site-specifically conjugate specific oligonucleotides oligos to the Fc regions of an antibody pair, minimizing the impact of conventional random conjugation on the Fab regions and maximizing the antibody's antigen recognition capability. When the specific oligos are brought into close proximity, they are ligated to generate an amplifiable DNA signal, followed by exponential signal amplification through qPCR. This triple specificity enables femtogram-level sensitivity even with ultra-low volume samples. VeriQuant experimental workflow 1. Specific antigen antibody recognition. Add 1 L of a sample, such as serum, cerebrospinal fluid, or cell culture supernatant, containing the target proteins. A pair of antibodies specifically recognizes the target protein to form a sandwich complex in a homogeneous liquid phase reaction. 2. Proximity of specific oligos. Within the sandwich complex, the specific oligos are brought into close proximity. Through the proximity effect, a specific ligation enzyme generates an amplifiable double-stranded DNA template. 3. Exponential signal amplification by qPCR. The resulting target-specific double-stranded DNA is amplified by qPCR. The protein concentration in the sample can then be calculated based on the CT cycle threshold value obtained from the target-specific fluorescent signal. 4. Multiplex detection. When multiple target proteins need to be detected simultaneously, simply add the corresponding specific antibody pairs conjugated with different oligos. Different amplification curves are generated by qPCR. The concentrations of different proteins can then be calculated based on their corresponding CT values, enabling multiplex protein quantification from the same sample. VeriQuant requires as little as 1 liter of sample, only 1% of the sample volume typically required for conventional ELISA. Sensitivity can reach the FG/mL level. It also provides a 5 to 6 log dynamic range, extending the quantitative detection range by more than 200-fold compared with conventional ELISA. At the same time, multiple proteins can be quantitatively measured from the same sample. VeriQuant immunoassay kits provide a powerful alternative to conventional ELISA and represent a new generation of protein quantification kits. With ultra-low sample consumption, high sensitivity, an ultra-wide dynamic range, and a simplified workflow, VeriQuant addresses key limitations of conventional ELISA, including high sample consumption, labor-intensive procedures, and limited detection ranges. It brings protein quantification toward greater efficiency and precision. We provide complete assay kits that create a streamlined workflow from sample loading to data output, helping researchers advance their projects with greater efficiency.